Corinna.
Okay, I share my screen first, I think.
Yep.
And do you see the full screen now?
Yeah, I do. Excellent.
Okay, perfect. Okay, great. Okay, yeah, thank you so much for inviting me today. My name is Corinna Breusing, and I'm a Marine Research Associate at the University of Rhode Island Graduate School of Oceanography. And I'm going to talk about mesopelagic animal microbiomes today. So for everybody who is interested in more details of this study, we recently published this paper that I'm basically going to present in this talk in ISME Communications. And this was a collaboration with Karen Osborne from the Smithsonian National Museum of Natural History and MBARI, Pete Gürges from the Harvard University, and Aspen Rees from UC San Diego. And before I go into the actual talk, I also would like to acknowledge the crew and the ROV pilots of the RV Western Flyer, Kakani Khatija, and the MBARI Bioinspiration Lab for making the sample collections possible, as well as the Bauer Core facility at Harvard University for preparing all the sequencing libraries for us and actually doing all the sequencing. And of course, also our funding bodies that made our study possible. Okay. So to just give you a little bit of a background about microbiomes or animal microbiomes. So microbiomes include essentially all kinds of microbial organisms, including bacteria, archaea, fungi, and protists that are associated, in this case, with animals. And they can have a lot of beneficial functions. For example, it can aid in digestion. So they are often involved in providing nutrition for the animal. They have also been shown to be important for immunity, as well as playing roles in animal development and physiology. But typically microbiomes also include, per definition, pathogenic agents that can cause disease. Increasing numbers of studies have shown that microbiomes are very important on an organismal level, but actually have more far-reaching functions that reach to the ecosystem scale and are also known to increase resilience of ecosystems, for example, to environmental change. So this is an ongoing area of study. And in the marine environment, actually most microbiome studies so far have been done on benthic invertebrates, especially corals and sponges, where there's less known about microbiomes of pelagic animals, and in particular mesopelagic animals, although those animals and their microbiota certainly have critical roles in the oceanic food web and global biogeochemical cycles. So for example, it's estimated that about 90% of the organic matter that's produced in the epipelagic is actually respired within the mesopelagic zone and then recycled into nutrients that can be used again for photosynthesis in the light flooded ocean. Mesopelagic animals also typically perform diel vertical migrations and thereby significantly contribute to the export of carbon to the deep sea. A lot of the animals feed on actually the tridal matter, which is often a very low quality and low abundant food item in the mesopelagic. So it's hypothesized that these animals actually have microbiota that help to extract very efficiently the nutrients out of, for example, the low abundant detritus and low quality, that particular low quality food item. But overall, it's very little is known about the composition of mesopelagic animal microbiomes, and it's also not really well understood which factors influence the composition, although there are certainly several factors that likely play a role. For example, the host taxon could play a role, probably the depth where the animal typically lives, the diet, and also the vertical migration status. And so, yeah, we wanted to basically look into this question a little bit more in detail. So we wanted to look at the microbiome composition and functional potential of the microbiomes for different mesopelagic animals, and then also understand the factors that might influence the composition and the functional potential. So we collected 11 midwater species from Monterey Canyon in California. And this just gives a brief overview. So we had 2 isopod species, 3 fish, 1 krill, 2 polychaete worms, 1 shrimp, the vampire squid, and a salp species. And we just recorded we collected them from different depths and then recorded whether they were vertical migrators or not. And so in total, we had 3 confirmed vertical migrators in this data set. That's the lanternfish, the skrill species, and the shrimp Eucergestis. So, and then we dissected either the gut tissue or whole body from these animals. We extracted the DNA, and this DNA was then used for 2 different sequencing applications. First of all, the 16S ribosomal RNA amplicon sequencing. So this is basically just targeting a marker gene for microbes to typically get an idea of the taxonomic composition. And we also did metagenomic sequencing, which is a whole genome sequencing for those microbiomes, mostly to get more information about the actual functions that these microbes might have. And so, yeah, we then use those data to infer the taxonomic composition of the microbiomes. We actually also use the data to get a little bit more information about potential diet of these animals, and we inferred the functional potential of the microbes. And so I'm starting with the microbiome composition based on the 16S marker gene that we used. And this just gives an approximate overview of the proportions of the most abundant microbial groups that we observed here, shown as on the class level. So a lot of the species had high abundance of gamma proteobacteria. We saw also quite a few bacilli and clostridia in some of the animals, and some of the isopods, for example, had a high abundance of campylobacteria, while other groups were relatively less represented. But when we actually looked on lower taxonomic levels, like more on the genus to species level, we actually saw quite a few more differences among the species, and also often among the individuals within species, and often saw that, for example, in most cases, only 1 or 2 bacterial microbial taxa were dominating the microbiomes. Yeah, so this is just shown because on the class level, because otherwise it would be too the figure would be too overwhelming. So we also did this analysis with the metagenomic data, for which we actually had, unfortunately, lower read depths. So we couldn't do this analysis for all of the animal taxa that we investigated. But for the ones where we could do this analysis based on the metagenomic data, you already see that this figure looks kind of different from the other one. So there are definitely some methodological differences in terms of the inference of the taxonomic composition. So we saw quite a few more bacterial microbial taxa that were in this other group, and also a higher fraction of actinobacteria based on the metagenomic data. So that's an interesting discrepancy that we would definitely like to investigate a little bit more, what kind of factors played a role in these differences. We also got some some of the first genetically informed data on the potential diets for those animals. And overall, the diet items that we inferred agreed pretty well with what was known in the literature already, and from, for example, stomach content analyses. But in some cases, they did not agree or were unexpected. So, for example, this Tomopterus worm here is known to feed on gelatinous zooplankton based on the mouth anatomy that it has. And it's And so we didn't actually see any gelatinous zooplankton in there, but just krill and phytoplankton based on the genetic data. So that was an unexpected result. Okay, so we also tried to infer, as I already said, what kind of factors could play a role in shaping the microbiome compositions. And so we used both the 16S gene amplicon data and the metagenomic data, and did a statistical analysis like a permanova, but also plotted these with a principal coordinate in a principal coordinate plot. And in those plots, we colored the data by host species, and the shape shows the diavertical migration status. And you see that in some cases, so for example, here this Eusagestis shrimp, the lanternfish, and also the krill showed pretty good clustering. So the individuals or the samples for those showed pretty good clustering by host species, while typically in the other species, we saw higher variability in the composition among the different samples. And the interesting thing about this is that all of the taxa where we saw this, what we call host-specific microbiome, were all vertical migrators, where the non-migrators typically had a higher variability among the individuals and less host specificity. So we also did this analysis, basically it's the same plot, but just colored by depth, and the shape shows the diet. We don't really see any effect of the diet in these plots. At least we couldn't see any, although this factor was significant in a statistical analysis, so explained relatively little. But you see a little bit of clustering by depth. I mean, it's not a super clear pattern. It partly overlaps with the host taxon, but we saw like a little bit of a relatively shallow group. Then maybe you can see here it's like a medium depth group, and then some deeper depth group. So some clustering by depth, and that was also confirmed by the permanova analyses that we did. And overall, host taxon and depth had the highest explanatory power, basically, for the microbiome composition. And so at last, we also looked at the metabolic potential. So we basically annotated the genes from the metagenomic sequencing and inferred what kind of pathways these microbiomes might have. This is just a snapshot from a relatively large figure in the paper, and just shows the carbohydrate and carbohydrate derivative metabolisms. You see here quite a few different degradative pathways. For example, they were able to degrade or potentially degrade all kinds of different carbohydrates or their derivatives. They had potential for fermentative metabolism, and also biosynthesis for different polysaccharides and so on. And overall, the metabolic diversity mirrored pretty well the taxonomic diversity and also the clustering that we observed. So for example, again, the lanternfish, the shrimp, and the Euphorsia krill were overall showing a good, so the individuals within those species showed a relatively good clustering together. So again, based on the host-specific microbiomes, they also had a more kind of, the individuals had a more similar functions for the microbiomes, while we saw usually higher variability in the microbiome functions for the other host species, which were mostly non-migrators, in a lot of cases also were mostly detritus feeders. Okay, sorry. So yeah, this was just showing the carbohydrate metabolism. What we saw quite a few other metabolisms that were present in the microbiome. So we saw a lot of potential for degradation of aromatic compounds and chlorinated hydrocarbons. A lot of the microbiomes had also potential for methanogenesis, which is an archaeal metabolism. Interestingly, we also saw potential for photosynthesis, which is interesting because in the mesopelagic, of course, the light intensity is not high enough to perform this metabolism. So we thought this might actually, so the microbes that had this potential were maybe not actual parts of the microbiome, but maybe second, like ingested phytoplankton or just somehow otherwise associated with the animal, but not actually real parts of the microbiome. And the last metabolism that was relatively abundant was formaldehyde assimilation, which is a pathway that is present in methanotrophic and methylotrophic bacteria and can often be used for example, amino acid biosynthesis. We also saw some differences among the detritus feeders and the zooplanktivores and herbivores that differed in terms of the abundance of, for example, nitrogen and sulfur reduction and also anaerobic hydrogen metabolism. Overall, most of the functions that we observed seem to have a digestive role. So a lot of potential for degradation of various compounds, but we also saw potential for biosynthetic metabolisms like production of vitamins, which certainly are important for the host animals. So just to kind of summarize this a little bit and interpret a little bit more what this means. So first of all, the host taxons seem to have the strongest effect overall on the microbiome composition, especially in the vertical migrators. Though when we compared our data, like the microbiome composition to seawater microbiomes from the same area that was published in a different study, we saw that all of the host-associated microbiomes were different from the actual seawater microbiomes. So there's definitely also in the non-migrators some kind of discrimination in terms of what microbes they take up from the environment. The interesting thing that we observed was that none of these mesopelagic animal microbiomes showed any sign of phylosymbiosis. So there was no phylogenetic signal in the data. That means we did not see that, for example, host species that are more closely related phylogenetically also had more similar microbiomes. So we did not see that at all. But by contrast, other ecological factors seem to play a role, like the vertical migration status, where we saw that the vertical migrators typically had these more defined, almost host-specific microbiomes, while this was not as common in the non-migrators. And that can have different reasons. So for example, it's possible that the vertical migrators have microbes that are inherited and not just taken up from the environment, and that would technically increase the host specificity of those particular microbes if they are transmitted from generation to generation, basically. It's also possible that the vertical migrators just have a more defined microbiome and just have microbes that have to tolerate the environmental fluctuations that they experienced during the vertical migration. And there could also be just differences in the host immune and physiological control among the groups. That's another potential explanation, as well as the detrital diet of the non-migrators. So a lot of the non-migrators were detritus feeders, and just because of the fact that detritus often consists of all kinds of different organic compounds. It's very unpredictable what it consists of. So maybe those animals have just a higher microbiome diversity to be able to digest the detritus items. Overall deaths seem to also have some influence on microbiome composition, though this was not always to be able to be disentangled from the host taxon effect. And so in the future, it would be interesting to, for example, sample the host species from different depths and then compare the microbiomes and see if they vary within the same species, but based on the depths where they were collected. So in this study, we only had each taxon from one depth. So we, as I said, we also got some information about the potential diet items, which was really cool based on the genetic data. But some of the items were actually pretty unexpected, like these crustacean sequences in the Tomopterus worm. Though it's possible that, for example, these sequences were observed because the species secondarily ingested crustaceans. So, for example, it's possible it ate, for example, a ctenophore that had eaten some kind of little crustacean or something like this. And that's why we picked it up, even though we didn't actually, even though it actually is not able to directly feed on crustaceans. And yeah, so overall, the metabolic diversity mirrored the taxonomic diversity pretty well. We saw a lot of heterotrophic metabolism or potential for heterotrophic metabolism and degradative metabolisms, which suggests that the functions of these microbiomes are probably in helping in digestion. And the highest metabolic diversity was observed in the detritus feeders, typically, or the non-migrators, likely because of the diversity of or the difference in composition of the detritus. We could not, of course, investigate all kinds of other factors that might have played a role in the or might impact in microbiome composition, like seasons. And the other interesting and last point was that there were some discrepancies among the methods and for inferring the microbiome composition accurately. And that could be due to differences in the read coverage. So for the metagenomes, we didn't have as many reads, but also the annotation databases could play a role, that there were limitations, or that some microbes might have been preferentially amplified. That can happen too sometimes, and might contribute to some of the differences we observed. And yeah, it's definitely something that should be addressed in a follow-up study. Yeah, so that's it for today, and thank you so much for coming and listening to the talk.
Thank you for giving it. It's great, amazing study and lots of information. I mean, if people have questions, again, raise your hand or put it in chat. I think I have just a question, if I can find it actually. Oh gosh. Would you say there's a difference, or that it could be interesting for in the future to figure out where the microbiome is kind of from? So if you take it from the stomach of the animal, or skin, or if there's a difference, or do you think that doesn't really make a difference? If you extract the stomach, for example, and then compare it to
Yeah, no, I think it will make a difference. So you mean investigating microbiomes based on tissue type? Mm-hmm. Yeah, I think that will definitely play a role. Just okay. Because they could have different functions in different parts of the body. So that would be very interesting, definitely. For some of the
I know it's not always feasible because I mean the animals are kind of small, but still,
Right. Yeah. So that's that's what I was about to say. For some of these, we were not able to dissect the gut tissue, for example, because they were just too small for that.
Yeah, totally. So. Yeah, I get it. Ali?
Yeah, thanks a lot. This was super interesting. I have a question that is a bit of a side question about the diet. So you're obviously looking for a big picture distinction between different diet items, but I was wondering, in theory, how fine into the taxonomy could you get? For example, when you were mentioning phytoplankton, could you potentially distinguish between different groups?
Yeah, it's definitely possible with the genetic data. We actually did this a little bit based on the 16S data. It just didn't show this now because wouldn't have fit into the table. But we saw a lot of diatom sequences overall, and then some other ones as well. Of course, with the genetic data, it's often difficult to interpret if you don't have comparative like stomach content analyses or isotope data at the same time. It's sometimes it was a challenge to, for example, distinguish between microbiome components, potential diet items, and then also contaminant sequences that we had to exclude and things like that. So
awesome, thanks. I'll check out the paper.
Yeah, we have much more information.
Thank you.
Yeah, if there's no other question, I think I would just end the call with this. And thanks again for both of you preparing such an amazing talk. And yeah, please go and check out both of their paper because yeah, that would be really cool. And thanks everybody who could join us. These videos will be on the YouTube channel probably in a few days whenever Adrian has time to do it. And I will stop the recording now.